All right. Great. Welcome, everyone. My name is Louise Pedrozella. I'm with BioMADE, and I serve as one of the senior program managers for education and workforce development here. We have been pleased to present this webinar series covering the projects that our institute has put out, the first and second project call from the beginning. We have a treat today because we have a project call from our first go and from our most recent PC2. So delighted to have everyone here. We can begin if my slide will advance. There we go. Just a little bit about BioMADE. Many of you on the line, I'm sure, are quite familiar. We began in 2021. We are sponsored by the Department of Defense. And we're certainly working to help develop and build the biomanufacturing bioeconomy here in the US. A number of things we do here you can see, but most certainly we're focused today on ensuring that there's enough workforce to meet the demand for the economy that is building. Noted here is our vision and mission. We're very proud, again, to be at the forefront of helping to develop this ecosystem and taking part with these projects and sponsoring these projects to help meet the demand in the workforce. So today, as I said, we have two projects. We will kick things off listening to about the project in Ohio with Amy Breeden. Then we'll take a brief pause. We'll throw it over to University of Texas, Austin with Kara Rogers and Katie Hansen, and they'll discuss the project ongoing there. They were part of the Project Call 1 down in Texas. And we'll hold any and all questions until after the presentation with Katie and Kara, so everybody has a chance to speak and have a good time letting you know what's going on. All right. I am going to stop sharing. I'm going to turn it over first to Amy, as promised. Amy, take it away and thank you.
All right, test this out. Okay, look good? All right.
Looks good.
So thank you, Louise. As mentioned, I'm Amy Breeden. I work at UES, and I'll be talking a little bit about UES in general, and then also our program that we've named Dayton, Maine. Right. So UES, we are in Dayton, Ohio. We've been in Dayton we were founded and been in Dayton since 1973. So we're actually celebrating our 50th anniversary this year. And we provide a range of R&D services and products, both at the corporate level as well as for customers. So one of our main customers is the Air Force Research Lab in Dayton, Ohio. We have over 300 employees with over 50% having advanced degrees, so a very technical rich company with 90% of our employees in Ohio. However, we do have some teammates kind of throughout the country, but we've been a part of Ohio's tech ecosystem for, since the beginning. We're very active in STEM education, as well as talent and workforce developments. So I'll touch on that a little bit more in a bit. And then very recently, we've announced that UES now has a private equity partner in a merger with Arlington Capital Partners. And so UES is now one of the 5 initial members of the Eclipse platform. And so we'll help continue to grow the Eclipse platform, and we're the lead member right now in their R&D sector. And so we'll still continue our work in Dayton, but we'll be able to kind of leverage our footprint in new growth areas. So we're very excited. UES specializes in a number of different areas, as you can see here from our technical divisions. So again, they range of various technologies from materials and processing all the way through bio and power and propulsion. So a whole range of different expertise. A lot of our bio work are in the bio and nanotechnologies division, as well as our integrative health and performance sciences division. And so some examples of biotechnology at UES in the bio and nano technology division, we have various different things dealing with biofuels, as well as biosedimentation and rare earth element extraction is one of our newer projects. And then on the integrative health and performance sciences side, we have microbiome research, a number of different projects developing or using biosensors, as well as some organ-on-a-chip technology. And so we definitely work with biotechnology in a wide range of applications and areas. And then in addition to R&D services, we also have a number of products and emerging technologies um, that kind of range the portfolio of from discovery through deployment, most recently with our element liquid metal inks, and then we also have had our RoboMet instrumentation. And so we have a number of, like I said, products in that pipeline. And then finally, like I mentioned at the beginning, we've been committed to STEM education and the next generation, again, throughout our history. We work with STEM education and outreach in a number of different ways, whether it's community support through like sponsorships and scholarships and whatnot. One of the main ways that we interact with the community is through mentorship and outreach. So we have a large number of student employees or internships throughout the year, and other programs that we bring in students for visits and tours. Our biomanufacturing summer camps, which we'll be talking about today. Mentorship is one of our big things. So not only having the students in the labs, but also things like iGEM team. We've helped mentor iGEM teams for I think 5 or 6 years now, and just working with various academic groups in the area. And then not only working with students, but UES is also committed to lifelong generational learning with programs for employees. And so with that, like I said, today I'll be talking about these biomanufacturing summer camps that we're putting on. And so we've titled this program Dayton Made, kind of play on BioMADE there. But it stands for Dayton Biomanufacturing Awareness and Discovery Experience. And so, like Louise mentioned, this was funded through BioMADE's EWD project called 2. And so this is our first year developing this program. And UES is the lead organization, and we also have a partner organization, Biopharmaceutical Technology Center Institute, or BTC Institute. And so the general project overview for this project is that we're developing biomanufacturing camps for underserved communities in the Dayton area. And this incorporates a number of different aspects. So first is active learning activities, really getting these students hands-on in the labs. As well as local industry and academia exposure. And then finally, incorporating specific kind of soft skill development. And I'll touch more on these in more detail in a minute. But that's the general kind of concept of these camps. And then the partners, both UES and BTCI, are bringing in our expertise as well as our networks to help develop this camp. And then we'll be producing a program and curriculum to the BioMADE community and just the community as a whole. And so the program kind of has 3 overarching goals, or main goals, sorry. The first one is to enhance biotechnology and biomanufacturing awareness in local underserved communities. Doing this a number of different ways. First off, through those hands-on lab activities. It's going to be very hands-on lab focus and really kind of getting exposure to those different techniques and skills. We also are bringing in a number of speakers or doing different tours. We're actually recruiting for this. I'll touch on this at the end of the presentation. But so that they can kind of see a breadth of different paths and opportunities and what's available in biomanufacturing. And then at the end of the camp, they'll be doing a presentation. We're calling it kind of a family and friends presentation or demo day. But they'll be kind of talking about something that they've learned throughout the camp and kind of teaching their friends or family or parents. And so that's actually a way to get awareness even further out in the community to, again, the families or community members that come for that. The second goal is to educate high school students on related local career and education opportunities. So On the education side, we're having a number of different recruitment or info sessions from various education, I guess, programs in the area, whether that's high school programs, community college, or four-year institutions. And so if they are interested in this area, kind of showing them all of the different avenues that they could pursue from the education perspective, and then also interacting with students from all of those different levels as well. And then on the career side, we'll be introducing them to various different career paths, but one of the main things is that they'll be doing a field trip to a local biomanufacturing facility and being able to not only see biomanufacturing at scale, but learn about the different career opportunities that are right in their community. And the third goal is to bridge the gap between current curricula and industry relevant skills. So first off with the hands-on lab activities, of course, with the various different biotech techniques. But also one of the key areas here is the softer power skills. So that's something that we've heard or we've seen with missing when we talk to industry. And so we're working with um, University Student Success Center to help us kind of give some targeted sessions on that. And then lastly, those end of camp presentations will also help develop some of those skills, whether it be like teamwork or just, presentation skills. And then underlying all of this is the idea of increasing self-efficacy. And so taking these students who haven't been exposed to this kind of work or this field and really being able to get themselves to see it as a possibility for them that they could pursue this path, whether it be in biomanufacturing or other STEM careers, but really increasing self-efficacy and the idea that they could pursue this. And so a little bit more detail about the camps. We're planning two-week camps each summer, this summer and next summer. So we have not actually held a camp yet. We're still in the I guess planning process. Each camp we're targeting having 12 students with 2 teachers and 1 to 2 student assistants. So it's a very kind of intimate learning environment. These will be hosted at Wright State University, which is a local university in Dayton, Ohio. And they'll be full day camps. So they will be spending the majority of their 2 weeks, at a college campus, kind of getting that experience as well. And then we're targeting underserved and financially disadvantaged populations. And so the 2 camps each summer, we have we're targeting 2 different exposure levels. These are all for high school students. But the first camp we're calling Biomanufacturing Explorers. This will be taking place in June this year, and this is for students who have little or no prior experience to biotechnology or biomanufacturing. And then we have our second camp, the Biomanufacturing Trailblazers, happening in July. That is for students who have some experience either with biotechnology or advanced biology, but they haven't really been exposed to the biomanufacturing aspect. And for both of these camps, we just require that they've passed high school level biology. And so we're definitely going to be exposing them to the basics and a little bit more advanced depending on the 2 camps. This year, they're going to be very similar. So this is an example of what our draft program looks like right now. Next year, they're probably going to be a little bit more different, but this year is going to be more about the execution of the content. But the program will be very similar. And so the first week is really kind of about the basics, introducing the students to the idea of biomanufacturing, and then all of the different, basic biotech techniques that are related to that. So aseptic technique, DNA, gel electrophoresis, microbiology, all the different kind of basic techniques and knowledge that they would need. And then we're kind of finishing that week off with, it's kind of a fun day, I think, but still somewhat related, is Biomaterials Day. So really showing them kind of the fun things you can do in that area and what's being done in biomaterials. Just to kind of close out the week on a fun note. And then week 2 will be more focusing on biomanufacturing and talking a little bit more about the kind of more advanced side, so bioreactors and scale up. We have a local professor who's going to be coming out and helping out with that. The industry field trip that I mentioned, so they'll be going and seeing again biomanufacturing at full industrial scale. And then in the lab, they'll be taking all of the things that they learned in week one, all of those basic techniques, and applying them to chromoprotein production. So going from beginning to end, and doing their own kind of mini biomanufacturing. Again, using those skills from the first week. And then the second week will end, or I guess the whole camp will culminate with that family and friends presentation that I mentioned earlier. And then one of the key things that we've really kept in mind while developing this program is affordability. So lunch, lab supplies, and a camp T-shirt will all be provided. But in addition, or more, yeah. So in addition, it's free registration, so there's no tuition involved. Free transportation for students who need it, as well as a student stipend. And so a lot of times in the summer, students will need to go or tend to get jobs to bring in some money during the summer. And so we wanted to really lower all barriers to participation in the camp. And so we've done that in this way. And so applications are currently open. We have a website, www.ues.com/DaytonMade, if you want to look at it. There's additional just information there as well. And so we've been collecting applications throughout the month. The deadline is the end of next week. And so we're really excited with the applications that have been coming in so far. And hopefully we get some more we will get some more, but we're really excited in what we're seeing so far. And then lastly, I want to touch on what I mentioned earlier, that we're in search of speakers. So we're looking for engaging speakers and diverse professionals to highlight various career paths in biotechnology and biomanufacturing. And we're looking to kind of highlight folks in a number of different ways, whether that's scientist profiles, so kind of just putting like mini bios inside of our lab manual or materials. But we're also interested in bringing in speakers in person if you're local, or virtual if you're not, or if you have a facility that you can give a quick virtual tour in or tour of. So if any of that sounds interesting, please either email me or fill out the form at www.ues.com/biotech-speakers. And looking forward to hearing from some folks about that. And so with that, that's kind of where we're at. Like I said, that we're having our first camp in a couple months, so we're definitely in crunch period and really excited with how things are turning out, and looking forward to giving an update once our camps take place. And if you have any questions or suggestions or whatnot, there's my email there. You can feel free to reach out.
Wonderful. Thank you, Amy. I'm very excited about these camps and for the students. And I have lots of follow-up questions myself, I know, but I'll hold on to that so we can have Kara and Katie discuss the project down in Texas. I'll turn it over. Thank you.
All right. Can everyone hear me and see the screen? Are we operating? Great. Excellent. Thank you all for the opportunity to speak to you today about our project with BioMADE that's happening at the University of Texas at Austin. And what we've been doing there is working very intentionally to integrate some biomanufacturing workforce development in scalable research experiences for freshmen. I'm Kara Rogers. I'm the director of a program called the Freshman Research Initiative, which I'm going to talk to you a lot about today. But I'm also joined here by my colleague, Dr. Katie Hansen, who is a research educator in our FRI program and is also the one to be developing a lot of the curriculum and implementing the activities that you're going to hear about in our BioMADE project. So I'll hand it over to her in a minute to discuss that. But I want to kind of give you the stage for what our project with BioMADE is, and it's really to build off of a successful program that we've had at UT for the past 18 years now, I believe, called the Freshman Research Initiative. This is the largest undergraduate research program in the nation, and we currently serve over 1,000 students every year in their first year on campus in authentic research experiences. And really what our mission is in the FRI is to make research not an extracurricular for students, but a co-curricular for students. We want them to be practicing and doing all these amazing research activities and gaining all of these amazing research skills that we know are important and enhance their learning outcomes along with their other curricular things that are going on. But we want to make it part of the coursework that they're doing, make it experiential learning, one of those requirements that really helps them progress and we know prepares them better for careers. At a traditional university, the research and teaching missions are largely distinct, and you have kind of your research faculty, the principal investigators are doing their kind of own research with postdocs or graduate students and staff. And then you have the teaching mission where you've got instructors and perhaps graduate TAs who are working with the students in bulk and trying to get concepts to them. And there do the two kind of meet. And really it is our goal in the FRI to remove this barrier. And we do so in kind of an integrated fashion where we bring this principal investigator with an instructional faculty member that we call a research educator together with the students to achieve a research mission. So I'm going to give you a little bit of an overview of how the FRI program itself works, just so you get a sense of what the student experience looks like, kind of at the base level. And then Katie will talk a little bit more about the specific biomanufacturing focused FRI experience that we've tailored for students. So just a little bit of jargon, because I always forget to say this. We call all of our undergraduate research groups in the FRI program research streams. And we have right now about 35 different research streams in the FRI. Our college at UT, the Natural Sciences College, is large and diverse. So we span a lot of things. We've got biology, biochemistry, chemistry, neuroscience, ecology, environmental sciences research, but we also have math and physics and astronomy. Computer science lives with us, as well as robotics. So we've got a lot of different fields of research that we encompass because we have a very diverse student body with a lot of diverse interests. And so we tried to find something that matches all of them. One thing that's kind of core to our structure in the FRI program is that each of the research streams is helmed by a principal investigator. So this is our research faculty member, tenure track faculty at the university who have their own research program. They have their own research lab or research group, perhaps, that, you know, they have postdocs and grad students, but we consider this research stream kind of an extension of their research program. And this really makes sure that we stay rooted in authentic research and that we are following novel research questions that have yet to be answered, but also have relevance in a field, in a scientific field. I believe kind of one of the special sauces to FRI success over its many-year tenure is our implementation of an instructional faculty position to support each of our research streams. And this is a teaching faculty role at UT, and we call these folks functionally research educators. And this is because they're PhD trained scientists. So they are researchers. They do research, they're experts in their field, but at the same time, they're also amazing educators, and they know pedagogical practice and know how to effectively communicate to students. And they have maybe the toughest job ahead of them because they have to take a really advanced scientific topic, research field area, and then teach students who are freshmen, mind you, who we don't enforce any prerequisites. We want all students to come and try things out. So they basically got to take all students kind of from different starting places, but essentially ground zero, get them ramped up in a very technical scientific research field, and then have them doing their own research by the end of it. So these folks are really special, and Katie is one of them, and you'll hear a little bit more from her in a second. Another really successful piece of our model has been the use of undergraduate peer mentors. So kind of in lieu of having graduate TAs for our FRI research streams, After students complete their freshman level experience with us in the FRI, we encourage them to return, and they can come back in these peer mentorship roles where they're able to co-teach and help that new cohort of students who is coming in to learn. And so they help train and also can continue their research. So this helps us get a little bit of vertical structure in our groups so that you've got kind of more senior students, more novice students, they're all working together, and we form kind of that research lab ecosystem. And then last but not least, of course, is all of the students who are enrolled in our course. And each of our research streams has something like 35 to 40 students who are starting their research with us in the FRI program in each of these research streams. I want to give you just a very short overview of what the experience looks like. So the FRI timeline is about a year long of students with dwell time with us. As I mentioned, this is a freshman program. We're really trying to get students at the beginning of their college careers so they can use this experience to grow off of and build their career path from what they've learned in the FRI. So in their very first semester, first time they walk on campus, we engage with them, we teach them some early research methods, how to think, critical thinking skills, you know, what do scientists do, and then allow them to choose which research stream in the FRI they would like to join. It's very important to us that students have some measure of choice and input into this because it's highly motivating for them to have ownership over the research field and topic that they join. Ultimately, students will join one of the research streams in the FRI program in spring of their freshman year, and they actually enroll in course. So they get a course credit for this. If it's a biology lab, maybe they'll get an intro bio lab credit for participating in the FRI research. And this first semester is a lot of, as I said, there's on-ramping. They got to figure out what is the research question that their research stream is asking. They got to learn how to look in the literature, get the context of the field, and then on-ramp into all the techniques too. They've done nothing. So we have to get them from very basic techniques, but all the way up to advanced techniques and advanced instrumentation. So we spend a lot of that time in spring semester kind of doing the on-ramp and getting students prepared. In their second year, there's an intervening summer time where students optionally can continue their research with us. It's not required, but we do function and have our labs open year round. So we do make that available to students if they have time in their summer to join us. But more commonly, students will just do the spring and then return in the fall semester for a second long semester with us, where they'll continue doing research with their FRI stream. They've got a whole semester under their belts, so now they're more able to be independent, ask their own questions, kind of do that process of lab iteration, do the experiment, it fails, what happens, try again, start the process over. So second semester looks a lot more independent for students. After they complete that sequence, what happens next? I already mentioned that we do encourage a number of students after they complete that sequence to come back and stay with the FRI program and continue working in our labs. They can continue their research, or they can become a peer mentor and help teach the new students, and those aren't mutually exclusive, so we actually hope they'll be doing both in some fashion. That's kind of working in a lab is always integrated teaching and mentorship of the new folks who join the lab. But we also hope that the FRI will be a springboard into other experiences. So we do want to send students off to faculty labs in different areas on campus to get a more in-depth research experience, and potentially also connect students with internships or other opportunities that may be more aligned with their career path. Maybe they're not considering going into research, or thinking about a medical profession or an industry position, to connect them with those opportunities. I do want to stress, though, our context for our FRI research is within a course, but it doesn't really look like your traditional lab course. And if anything, we look more like research labs than we do a teaching lab. And we do that very intentionally because we want students to model and see what it's like to be operating as a practitioner of their field. And so Katie is going to talk about that kind of in the context of biomanufacturing and intentional efforts there to make students feel like they're in a biomanufacturing space. But because we function more like research labs, you're not going to see us really having like big lecture times. We'll more see students in small group meetings or coming in and out of lab. Don't really have kind of set lab blocks so much, as we've got labs that are open, students are coming in and out as their research needs dictate. They're working on advanced techniques and instrumentation, doing their own analysis. And you won't see things like final exams because we really put more preference on students doing what scientists do. And so they will be doing scientific communication, like writing manuscripts or presenting posters, and those sorts of things that an actual scientist would do. And all of this to say to great success. In our 18-year history, we've had over 500 FRI students who have been co-authors on publications or on presentations at kind of the national conference level. But personally, for me, one of the things that I think is the most impactful for students is the community that it establishes and forms. These students are joining in their first year on campus, and it's a large campus. So we make it feel small and give them a community that they can identify with that is really rooted in the practice of being a scientist. So it helps them establish that kind of scientific identity while also having that support of a community and social aspect. And they're getting long-term mentorship from both peers, as I talked about, but also professors and faculty. They're meeting faculty that they work with closely right off the bat, their first year on campus. And this is a very intentional part of our kind of recruitment and retention strategies here at UT. More than half of our students we identify as kind of being from at-risk backgrounds or underrepresented groups in STEM fields. So some other outcomes, aside from kind of research productivity, in our many-year history, we've kind of followed what has happened to students. There's a lot that they build on the psychosocial side of things, their perceptions towards STEM and their own identity in STEM and belonging, and how they feel about self-efficacy and the ability to achieve these things. On kind of a very measurable output end, we also know that FRI students are statistically more likely to graduate on time with a STEM degree. And then this last piece here, career skills. I like that Amy used the term power skills because I always think about it in that way too, instead of soft skills. But all of those communications, leadership pieces, technology literacy, data literacy, a lot of those things that are important to companies, students are building in the FRI by doing research. But where we're kind of taking this project is we really want to build some technical transferable skills for students to take into industry. And this is where our BioMADE project has kind of grown. And so seeing a need of some growing biotech industry in Austin, we really wanted to take the stream model that's been successful so far and put in specific industry workforce development and like biotech biomanufacturing space into that stream and build more process skills and see what that would look like in the FRI. So Katie's been growing out that curriculum for us. We've also been kind of leveraging other conversations that are happening right now at the same time. We have a local community college, Austin Community College, that's been doing biotech preparation and certification for students for years and are great at it. So we've been leveraging their expertise a lot and really leaning into this kind of national conversation on career preparedness and skill development and credentialing. And I think this serves us well. So now I'm going to hand it over to Katie and she's going to tell you more about how we've implemented things. Katie, you're on mute.
All right, there we go. Can you hear me?
Yes.
Yeah, okay, great. So the students in the biomanufacturing stream are embedded within our PI, Dr. Brian Davies, within his lab for 2 semesters, right? So they do their spring and then in the fall. And so I'll kind of walk through what that looks like for them a little bit. So the research that they're working on is to optimize a novel E. coli secretion system for small proteins and biotherapeutics. And this was just recently developed by a postdoc in PI Davies' lab, so it's brand new. It's just about to be published, so it's brand spanking new stuff. So through the course of their research, students are receiving training in GMPs, upstream, downstream processing, and then they'll also have the opportunity to test for skills certifications at the end of their experience. So we are also reaching out to local biotech companies to try to set up some infrastructure to create new internships and networking opportunities for our students. So you can see 2 of our student cohorts here. So FRI is a program that starts in the spring. So in the fall, we piloted just a really small group of 20 students to try to kind of trial some of our curriculum, and get some early feedback on that. And then we did a full-blown stream started this spring. with 40 students. So our current emphasis in research is kind of on the secretion system. We also have another PI involved who's Dr. Andy Ellington, and he'll be kind of feeding in some other projects in the future. So we'll have kind of multiple things going. Do you mind switching the slide, Kara? All right, thank you. So here are our students. You can see them doing all the things that they do. So when they arrive in the lab in the spring, as Kara was kind of mentioning, very few have had any lab experience at all. So we have to start from scratch. And so we'll, first teach them how to pipette accurately, because everything they do later won't matter if they can't do that first thing really well, right? So this spring we actually experimented with a pilot that's being developed by our biochemistry colleagues here on some micro certifications. And so we give our students the opportunity to get a micro certification in pipetting. And so they had to, in order to pass, they had to be able to pipette, things like water, but also really hard things like really viscous glycerol and bubbly protein solutions. And then that culminated with a smart check at the end. And so the goal with this was not just to make sure that they can pipette well, but we wanted to expose them early to the idea of skill certification, because when they're finished with the program, they'll have the opportunity to pursue some of those through our collaboration with Austin Community College. So we just wanted to kind of put that idea in their heads early. So we spent about 6 weeks of the semester teaching these basic skills, including, preparing solutions, sterile technique, learning how to use some of the more advanced equipment in the lab, like plate readers and gel imagers and things like that. And then across the next 4 to 5 weeks, they start preparing for their individual research projects. And so this is a really long process, because for them to do meaningful research, they have a lot to learn, right? And so we'll dissect journal articles on, small proteins or biotherapeutics. They'll, dig into the Davies research paper on the secretion system that they're using. And this gets them kind of comfortable, not only with the peer-reviewed literature, but it also allows us to kind of teach these important things that they need to know about the structure and characteristics of proteins. How secretion works in bacteria and how protein production can work. And so we kind of combine those two elements for about 4 to 5 weeks. And so we don't really have a lecture session, but we have a meeting once a week where we do those types of things. And then in the lab during this time, they're actually working to build that secretion system. And so it's a 2 plasmid system. And so one of the things about 2 plasmid systems is a lot of times the cells don't really like necessarily taking up that second plasmid very much. And so it's sometimes a little bit hard to get in there. So while most groups are successful, a few are going to fail. It's just the way it works. It happened to any of us in the lab. Sometimes it just doesn't work, right? But this starts a really important evolution in the way they think about and how they approach their time in the lab, right? So it changes from, these expected guaranteed outcomes to having to troubleshoot and figure out what went wrong with their experiments. And so it's super fun to watch, because this is where their deepest learning occurs. And you can also see them transform from being a science student to being a scientist through this process. So it's actually pretty fun to watch. And it's kind of one of the best things about working with freshmen is they don't realize what they can't do yet. So they will dive in and try anything. And for example, if you look at this image here, you can see in the second row, those are our freshman students learning how to pour polyacrylamide gels, which is something neither of my graduate students knew how to do. So if you aren't super familiar with these, they're something that we use almost daily in protein labs, but they're super easy to mess up. And so a lot of labs tend to purchase pre-made ones. But, like I mentioned, freshmen will try anything, and they were really successful at it. So it was a little win for us. Do you mind hitting the next slide, please? Thank you. Okay, so the focus of the research in our lab right now, as I mentioned, is on characterizing and optimizing this novel secretion system that was developed by one of Dr. Davies' postdocs. And so in the lab, what they found is that the system will secrete almost anything, and the only major limitation is the size. So it needs to be about under 116 amino acids. So that it creates a lot of opportunity. So our goal here is to try to scale this system up from a shake flask into a 5-liter fermenter. So to do that, we have to have, a better understanding of, growth conditions for optimal secretion, like pH, type of media, temperature. And we also have to understand a lot of basic questions like, how long will it secrete protein for? What are the impacts of multiple inductions or feeding? So these are relatively simple questions, but they're super important. They need to be answered in order to make this, viable at a larger scale. And so this is one of the ways where our students can, come in and how they can, add value to the faculty lab, because they can find answers to some of these questions that maybe the faculty lab doesn't have time to do. So the students will start by choosing a project that they're interested in. And then they will design their own experiments by converting a standard SOP for this system into a modified procedure that's specific to their questions. And this is really, really hard. We've all done it. It's very hard, right? So they just now finished conducting their pilot studies to see if their procedures would work last week, and the success and results were about as varied as you would expect them to be, right? Some worked really, really well, some not at all. But again, it's another opportunity to target those super critical troubleshooting skills that are really important, right? So they're going to continue kind of honing these procedures and expanding their research beyond the pilot as they move into the fall. So currently the types of proteins we're working on are mostly alpha bodies, which are small binding proteins, but we're also going to be adding a few biotherapeutics this summer. So as part of the FRI program, as Kara mentioned, we offer several summer fellowships across the summer for students to participate if they'd like to. And we'll be having 4 students do that this summer. And so their focus is going to be on the scale-up process from the shake flask to the 5-liter desktop fermenter. So ACC, or Austin Community College, has loaned us an ancient but functional fermenter that we are ridiculously excited about. And so this is going to allow our students to kind of experience and troubleshoot this scale-up process. And this spring, we've actually been working with the biomanufacturing instructional staff at ACC. They come on site to UT, help train us, help us find all the parts, get it all ready. And we're hoping to do our first run, trial run in the next couple weeks. Do you mind hitting a new slide, please? Thank you. So I want to call your attention to the bottom row of this picture. So last week we had our large undergraduate research forum with more than 500 students presenting their research. And so our first semester students, you can see in the first 2 pictures, they all work together to present their research at the forum. The second semester students you see on the right presented their own posters with the results of their independent projects. And so it's a super exciting event. And the excitement is just palpable when you're in that room. It's pretty amazing. And then I'll do the last slide. I can see we're running a little bit at a time. Okay, so in this diagram, you can kind of see our project overview and the different stages we're in. So we began kind of from scratch and setting up a new lab for protein research, and BioMADE funding is what made that possible to purchase many of the new pieces of equipment that we needed to do this. We did a trial run of our curriculum in the fall. We modified it through the spring, and then Dr. Linnea Fletcher from Innovate Bio is helping us evaluate our curriculum, identify gaps, and that will be kind of a process that continues on through next year and the end of the project. And so I think that's about it.
Yeah, I just want to end here and say, the BioMADE partnership that we've had has really kind of launched us into this ability to tap into synergistic activities, which are really growing in the Austin area. We talked already about our really fruitful partnership with Austin Community College, but in addition to helping with us with our curriculum, they've been helping build the fermenter out for us and make sure that's up and running. We've been talking about pipelines for our students to get certifications through ACC and launch them into lab placements and industry. But there's a variety of other folks that we work with. We're really engaging kind of with biotech companies that are national, but also local. We have a great partnership with Thermo Scientific here We just went and toured their biomanufacturing facility, and they came to this poster session that Katie mentioned, and actually sponsored our poster session that we had this past week. And then there's a lot of other conversations kind of on the government and defense sector that are also happening in the Austin area. So we've got an Army Futures presence here. There's also Army Research Labs South, and they actually have a physical presence on UT campus. And so we've been kind of engaging in also all of these DoD conversations around biotech and biomanufacturing workforce development, trying to figure out how we can work together to create and serve everyone's goals, but create cool new opportunities for students to train them in workforce skills that can be useful. So this has become quite an ecosystem here, and all to BioMADE's support. So last, I'll just leave us on our website. So please, if you want more information, head there. We have a page for our biomanufacturing stream and our contact information there as well. So thank you all.
Oh, thank you both. That was a great presentation. I want to be mindful of our time. I know we've got just a little less than 15 minutes. We'll open it for questions. And Chris, I knew you were going to be the first question out of the box. Go ahead, Chris.
Hey, I actually have a question for each of the presenters. So the first one's for UES, and I was curious if you can talk about how you are going to evaluate and come up with who gets to come if you have a lot of applications coming in. And then if I can tell you about the second presentation from UT Austin. I was curious if you found that having the research, this research institute or whatever it was called, the freshman thing, if you're seeing a reduction in students, in student attrition. I'm just curious if that has been looked at. So those are my two questions.
Amy, you want to kick it off?
Sure. So thank you for the question. We've talked about that some, but I think we do need to look at it closer now that we are getting more applications in, especially for the Explorers Camp. And so some of the things we've looked at is we have a sponsor form that we're sending to recommendation. That's the word. I'm like recruit now. So we're asking for like a science teacher that we can reach out to send the sponsor form that they can kind of give their input as to whether, especially with our camps being very lab heavy, if that's kind of the environment that the students would thrive in. And so we'll take those forms into account. We also had some short answers as to, why do you want to do this? So looking at their essays, and then also keeping an eye on the our target audience, right? So we need to talk about this more internally, but whether it's kind of a making sure that we're hitting those underserved students first. So we're still kind of working through some of that selection criteria, but that's some of what we're taking into account.
Thank you, Amy.
And I thank you for the question about reducing attrition in STEM. So I'm going to say it differently and say the impact of participating in the FRI program on persistence in STEM. And we do follow this closely. We do a lot of metrics and tracking. We have 1,000 students, so we got all the data. And we've got a lot of students who aren't participating in this. So we have really good comparison groups that we can look at. It's definitely clear that we have increased persistence in STEM. And so I did share very briefly a metric that on-time graduation rates, especially in STEM, are much increased and statistically with students who participate in the FRI program. So it fluctuates kind of year to year, and our college actually has done a great job doing other things to support students staying in STEM fields, but it's something around the ballpark of about 15% increase in students who participate in FRI continuing on time to get a STEM degree. So yes, we hope that they're staying here and enjoying their experience and feeling like they are scientists, right, by doing these things hands-on and early. Thanks.
Thank you, Kara. Bruce, your hand is up.
Yeah, so briefly, it's actually a follow-up, and I would have taken my hand down. But in terms of long-term effects, on career choice and success? Do you have metrics on that? Are you able to track people beyond UT to either Amy or Kara?
Oh, this is just, this is the holy grail of tracking these experiences. And Louise and Katie and I had talked about this recently too, because it is the hardest thing for us to follow and like where students are going. We have graduation surveys. And again, one of the benefits of UT is we're huge. So even when only 10% of students answer surveys, it's still a lot of students, and we get some information. So we do have some indication about where students go career-wise, and that FRI is having positive impact. But I think we need to figure out a better way to view these long-term tracking pieces. And also, our hope is to kind of do some more intentional placements too, and like put people in internships and in careers and build those pipelines out a little bit more. And we haven't done that, but that's kind of, I would say, the next phase. We're really getting the stream up and running. And then it's kind of like launching and what students do post to stream. That'll be kind of our next focus. But I wish we had better data to share on that, and something I would.
Yeah. Well, I have the same issue, and we don't have ownership of our students. We train faculty. So I'm actually very interested in coming up with a way to do this nationwide, maybe for more than just BioMADE, but for all programs. That's sort of the holy grail. But I think some way of getting data on people and what actually happens is probably possible. And I know everyone's interested. I was just curious about what you were able to do. Thanks.
One of our challenges is the immediate after UT or college experience is not always what the student's career ends up being. So we've got gap years. We have students who do something just for a couple months before they go into their ultimate career, and that time period is actually very challenging to overcome in the data collection. So.
Yeah, but thanks, Bruce. That is something that Kara and Katie and I have been talking about and trying to figure out how we can do that. Other questions from folks? Well, I have a comment and a question. Amy, love the Dayton BioMADE. I mean, the Dayton MADE name. I mean, yes. Wonderful. You recognize this, Amy. Transportation is a huge issue. We can offer camps and opportunities, but to get the students there sometimes, can you get into the weeds a little bit about the transportation arrangement? I'm curious about how you're going about that.
Yeah. So we are wow, my brain keeps pausing there. So we're setting up a few different central pickup points. So we're targeting 3 pickup points that we've kind of the 3 that we offered are kind of strategically placed near or like around Dayton in the different areas and near the schools or the communities that we're targeting. And so that's the idea is that we hope that they could get to these central pickup locations, but if that doesn't work, then we've told them to reach out and we'll make something work. And so with the application process, we like I said, proposed. We put out 3 different options. We've actually I want to say about 50% of our applicants have taken us up on the transportation offer. And of those, they've really focused on 2 of those pickup locations. And so that 3rd one, we're going to work with the students who none of our current options work for them. We'll work with them to kind of find a better 3rd option, if that makes sense. Or we'll work with them to figure out, how to make it happen. But the general idea is that we'll have these central pickup locations, and so we're working with a transportation company that will do that for us, or do the transportation for us. And then we're also using them to go to that field trip. So that'll be everybody kind of going all at once for that.
That's great. Yeah, that's what I wanted to know. Were you going to work with some, a company? Were you going to Uber the kids? How is it?
Yeah, we're working with a company. Yeah.
Oh, good. Okay. Kara, I thought I saw you put a comment in the chat.
I did. I was just really relating to Amy's struggle because we also do some high school activities, and it is hard figuring out transportation issues to make these things accessible. So I was just saying, if you learn some best practices, we'd love to hear, because we've kind of abandoned some of those efforts just out of not finding any real good solutions that worked well for us. So it's tough.
Yeah, thank you for considering, those wraparound services for students to get them where they need to go to support them as needed. We're running short on time here. Tom came on camera. Tom, question.
Yeah, I have a really great question. Based on the information that you all provided here, Kara and Katie. This is, I guess, a direct reference to the partnership with Austin Community College. I think that this is fantastic. Good to see the 5-liter bioreactor up and operational. Huge jump up in participation for students in the current cohort. And I'm a little bit concerned about access to equipment for 1 5-liter reactor. So I just wanted to tap your brain and ask you what your vision for potential expansion of this is, and maybe what your perceived need is for the equipment to get more people practical experience working with those bioreactors. Equipment shortages, any?
I'm going to let Katie answer this, but I also know she is about to go start a run at 1 o'clock. So I don't even know if she may have to jump off. Katie, are you there and you want to say anything? Otherwise I can answer. Okay, yeah, she is going to use her giant autoclave to get the fermenter up and running literally at 1 o'clock. So I hear your point. I think what we've been focusing, because the fermenter isn't even operational yet, we're doing our tests right now, is we've been focusing a lot on kind of benchtop expression and fermentation, in small scale right now, to get students kind of trained up in skills. I think once we get this system up and running, we're going to be able to see, but it's pretty onerous to get it set up. So I think we're going to have to do this in groups and kind of very intentionally staged times, because we have to autoclave the entire system and make sure it's completely sterile before we start the run. So I think this is going to have to be something that we do kind of in groups, and that's not uncommon in FRI. We kind of arrange our students in groups. So I see that being a lot more in our kind of second semester for students. They spend the first semester really gaining their own hands-on experience with sterile technique, doing small scale fermentation in flasks. And then in second semester, that'll give us a little more space and time to spread out larger fermenter runs, perhaps in groups, so that students can get access to it. We'd love the fermenter just to be constantly running all the time and be cranking out stuff. And I just have this vision that we're making stuff for the research labs and just churning it out, and it's always going. And I hope we can get there, but we just haven't done it enough yet to see what it's going to look like on the practical end.
I just wanted to make the comment, because with the portfolio that Louise is overseeing for BioMADE, we do have a number of our members who are building do-it-yourself bioreactors, tabletop bioreactors, things that really can help to democratize the technology.
Oh, yes.
And get the education out there. And so we encourage a lot of synergy between our programs. And you'll see that we're all one big network here. So resources are plenty. And so
awesome.
More room to talk.
I agree. And I think we want to try to do things that are going to be relevant to students as well once they get into industry. Unfortunately, the fermenter we have is a little bit old. And so we're trying to think about ways that we can get students kind of hands-on stuff. What will they be doing when they get to said company? And make sure we're trying to get them that exposure instead of something that wouldn't be used any longer. So happy to make connections and learn from other folks in this group.
Okay, that's great, Kara. And we can hook you up, because I know you're going to be at the member meeting next week. So lots of networking to be had there, and can turn you on to resources available for the students. I want to thank everyone for coming today. Really wonderful presentation, so informative. And this is, again, our final webinar for this series. But we do have our project call 3 out and about. We hope to fund some projects and hold these webinars and these informational sessions again here. And I just, again, thank you all very much. Hope to see you at the member meeting next week. Take care.